Hey everyone, welcome back to Windows Seed Studies. I hope you all are doing well. So, we were learning about cytoskeleton. In the previous lecture, we had an introduction about cytoskeletal elements, the different type of filaments that make up our cytoskeleton. So, in this lecture, we are going to learn about microfilaments, which are also known as actin filaments in detail. We will be learning about the formation of microfilaments, the different types of factors that affect the formation, the accessory proteins, the remodeling of actin filaments, and what do actin filaments do. So we already know that the microfilaments, they help in maintaining the cell shape or the structural integrity of cell. So they are found predominantly in the cortical region, means the cortex, the area or the cytoplasmic area which is found beneath the plasma membrane. So actin filaments are predominantly found in the cortex region. So due to this, the cell shape is maintained. So the cells which have a very distinct shapes like nerve cell or epithelial cells, we can find those actin filaments in the cortex region. So microfilaments, which are also known as actin filaments because they are made up of a protein known as actin. Okay, and the monomeric form of this actin filament, the monomer that makes up this actin filament, it is known as G-actin or globular actin. Okay, so the name suggests that G-actin is globular in shape, but it is not perfectly globular. Okay, so it is a kind of polar structure which has a which has a structure like this. So which has a barbed end and a pointed end. So the barbed end is also known as the positive end, while the end is also known as the negative end. So G-actin, it is not perfectly globular. This monomer is a polar structure. So when multiple G-actins, so they come together, they polymerize, they form F-actin. Okay, so we are going to learn about the formation of filamentous actin. So let's take a look at this diagram. This is showing the molecular structure of globular actin. So this is a globular actin. So globular actin has a distinct property of being able to be associated with ATP or ADP. So this is this is the site where the ATP can bind. So this is globular actin. So this is the N-terminal end. Okay, and this is the C-terminal end. So the barbed end is also known as plus end, while the pointed end is also known as the minus end. So here you can see this is a filamentous actin. So this is actually a right-handed helical structure where the two protofilaments, they are coiling up onto each other and they are forming a twist of the 37 nanometer. So this is how a filamentous actin looks like, and this is how the filamentous actin looks like under the electron micrograph. Okay, so let's now learn about the formation of the actin filaments. So this is a very step-by-step process. So this process occurs in three steps. The first step is known as nucleation. Okay. So nucleation, as the name suggests, means formation of nucleus. So in the case of F-actin or actin filaments, we call nucleus. Nucleus is actually a trimer of G-actin. Trimer, or you can say oligomer, because it doesn't necessarily mean trimer, but the basic requirement is a trimer should be formed. Three G-actin molecules should come together to form a trimer, and only then the process of forming filamentous actin or the process of polymerization of G-actin starts. So this is a kind of rate limiting state, okay, or rate determining step of the whole reaction. So once the nucleus is formed, now the whole reaction will speed up because formation of nucleus is going to determine the rate. So this is a crucial stage. After that, multiple G-actin will come together. They will start polymerizing. So multiple G-actin molecules will be added to both ends, the positive end as well as the negative end, and that phenomena is known as elongation. Okay, let me show you a schematic of the nucleation. Okay, so here you can see this is one molecule of G-actin. So this is G-actin. Okay, so when the other molecule of G-actin attaches itself to the pointed end. So this is the let's say this is the barbed end, and this attaches itself to the pointed end. Then the third the third G-actin molecule comes together and binds to the dimer. It forms a trimer, and this trimer is known as the nucleus. And the formation of this trimer is known as nucleation. So once trimer is formed, the other other G-actin molecule binds to this nucleus, and now the polymerization will start and elongation will be seen. So when the G-actin molecules come and bind to other G-actin molecule, it binds at an angle of 166 degrees. Okay, so they do not form a straight linear axis, so they do not attach themselves on a 180-degree axis, but rather they attach themselves on the 160 degrees. Okay, so this is how the process looks like. So first a nucleus is formed, the trimer of the G-actin, and then the elongation is seen. So G-actin molecules are added to both ends. But here's a catch. The rate of addition of G-actins towards the positive end and the negative end differs, because this is due to the conformational differences between the positive end and the negative end. That is why the name to the barbed end it is also known as positive end, because it is a fast growing end. It is also known as fast growing end, and that's why the the nomenclature positive, because it is fast growing, because the rate of addition of G-actin is higher. So rate of addition of G-actin, okay, at positive end, it is higher than the rate of addition of G-actin at negative end. Okay, that is why negative end is also known as slow growing end. So, during the elongation process, the G-actins, they are added to both ends, positive as well as negative. But the polymerization rate, or the rate of addition of G-actin at the positive end, it is higher than the rate of addition of the G-actin at the negative end. This is due to the conformational differences. Okay, so let's say that this is how the trimer was formed. Okay, and now after the formation of trimer, the G-actins will be added to both ends. So this is the positive end and this is the negative end. But the rate of addition of G-actin at the positive end will be higher than the rate of addition of G-actins at the negative end. Then comes the last stage, where the equilibrium is reached, because this is a reaction after all. There will be a stage when the rates, the rate of forward reaction and rate of backward reaction, they both will become equal. So in this case, what the reaction is? The reaction is polymerization, right? This is a polymerization reaction. So there will come a stage when the rate of polymerization will get equal to the rate of depolymerization, and that stage is known as the equilibrium stage, which is also known as steady state scenario. Because in this scenario, it looks like nothing is moving, right? So the reaction is not proceeding in any direction. So let's say that there are two reactants, A and B, and they are reacting to form a product C. But this reaction is reversible. So when a stage when the rate of formation of C and rate of formation of A and B, means the rate of forward reaction and rate of backward reaction, they both becomes equal. Okay, at this stage we call the stage as the equilibrium stage. So during this stage, it looks like the reaction is not proceeding in either direction. Neither the formation of C is seen, neither the like breakdown of the C to the A to B is seen. So it means the rate of both of the reaction becomes equal. So it looks like we have taken a screenshot of the reaction. We are taking a snapshot. It is not proceeding in any direction. That is why it is also known as steady state scenario. It doesn't look dynamic. It looks like a steady state. So in the case of the formation of actin filaments, so this is a polymerization reaction, right? So when the rate of addition of the G-actin will become to the rate of removal of G-actin. At that point of time, a steady state scenario will be reached. So in this filament, so now let's say that this filament, so F-actin, okay, well, G-actin, it formed trimer. Okay, that means the three molecule of G-actin came together to form a trimer, which is also known as nucleus. And now the multiple G-actins will be added. And let's say that it reaches to a certain length, the length which is sufficient to call it as an F-actin, the filamentous actin. Once this sufficient length is achieved in the cell, it will reach to the steady state scenario. Means that now rate of addition of the G-actin will equalize to the rate of removal of the G-actins. So even though the G-actins are being added to positive end as well as to the negative end, due to increase in length of this F-actin, certain G-actin, because this is a dynamic process, right? So this is a reaction. So it has both of the sides, the left hand side as well as the right hand side. So the G-actin to F-actin formation is reversible. So this is not only one direction reaction, but rather this is something like this, that G to F. Okay, so multiple G-actins are going to form a filamentous actin, but at the same time, certain G-actin molecules are being removed. But what happens is that in the elongation stage, the critical concentration means which is represented as CC. Critical concentration means the concentration of freely available G-actin, it is high enough, and again the length of F-actin is small enough to ensure that polymerization rate is much higher than the depolymerization rate. At the end, we say that the polymerization is seen at the both ends, positive ends as well as the negative end. But after a sufficient length of F-actin formation, like F-actin has sufficient length, after that we see that the rate of depolymerization increases, and it increases to such level that it equalizes to the rate of polymerization. So this is what we call as the steady state scenario. So rate of polymerization equals to rate of depolymerization, and that is known as the equilibrium state. Okay, means the number of G-actins added will be equal to the number of G-actins removed. So this is how in the three stages the formation of F-actin is seen. So we can also represent this graphically. Okay, so on the x-axis, I'm plotting time, and on the y-axis, I'm plotting rate. Okay, so the graph, so this on the y-axis, I'm plotting the rate of formation of F-actin. Okay, so the graph somewhat looks like this. Okay, so this is known as lag phase. This is known as log phase, the exponential phase, and this is known as the stationary phase, right? So first is lag phase, which is seen during this the nucleation happens. The nucleus formation takes time. Like let's say that in this cell, the G-actin concentration let's say the concentration of G-actin means the critical concentration is 1 micromolar. Okay. So now what means we have sufficient amount of G-actin in the cell. So now multiple trimers of G-actin will form. So this will take time. So once sufficient amount of trimers is formed, now to those trimers, the other G-actin molecules will start adding. Okay, and they will start forming F-actin. So this is how the nucleation takes time, and that is why it is represented in the lag phase. So it takes time. You can see it is slow growing phase. Okay, and the second one is the exponential phase, which is also known as log phase, when where the rate of formation of F-actin increases rapidly. Okay, it increases exponentially due to sufficient amount of G-actin, and that is known as the elongation phase. So log phase in the graph here shows the elongation phenomena. Why when and once certain time has passed, the length of so sufficient length of F actin has been reached. Now comes the stationary phase, the steady state scenario when the equilibrium is reached. Okay, so then comes the steady state scenario. So this is how the graph looks like. Okay. So, but why does this happen? So there's a term which is also used to refer the steady state scenario, which is known as actin treadmilling. Okay, so actin treadmilling is nothing else but the other term which we use for representing steady state scenario. So why does this happen? So actually, let's say that this is a F-actin, and this is the positive end, and this is the negative end. So the critical concentrations here means the concentration that are required for the polymerization of G-actin to F-actin at both of the ends, the positive end and negative end, it is different. Okay. So let me try to explain this in a different way. So let's say that this is G-actin. Okay, so here you can clearly see G-actin is a polar molecule. But when this G-actin is going to be, included into the F-actin, the conformation changes. Okay, and this is the G-actin subunit, which is going to be embedded in the polymer, the F-actin. Okay, so due to the differences between the positive end and the negative end, the conformational differences between positive end and the negative end, the rates by which they are going to be added, or the rate of polymerization at both of the ends, will be different. So when the G-actin is being added, so let's say that this is the polymer of length n. Okay, and now I'm going to add one G-actin molecule. Okay, and it will form a polymer of length n plus 1. Okay, so we already know that this is a reversible reaction, right? So let's say the rate of addition of the G-actin, it is known as K on, and rate of addition of the G-actin, it will be represented as K off. Okay, so the rate of dissociation of the polymerization, so that is we refer as the KD, the dissociation constant, which is exactly opposite to the equilibrium constant. It is also represented as K off, this is the depolymerization rate upon the polymerization rate of the subunit of the G-actin subunit. So this equalizes to the critical concentration. What is critical concentration? Critical concentration is the concentration of the G-actin which is freely available in the cell for polymerization. So theoretically, so like by equation, this is represented by the KD, okay, dissociation constant, the rate of the depolymerization upon rate of the polymerization. So what happens is that even though the polymerization rates, so we already discussed that the polymerization rate means K on will be much higher than the K on of the negative end, right? So the polymerization rate at the positive end will be higher than the polymerization rate at the negative end. But the ratio of the depolymerization to the polymerization, that will be same, because this equalizes to the critical concentration, the concentration of G-actin which is available to cell, and that is equal in the, that is equal in the cell. So like this cell, in this cell, the cytoplasm, the concentration, let's say it is 1 micromolar. The concentration of G-actin is 1 micromolar, and that, let's say that is the critical concentration. So this is fixed. This is fixed value, and that is why the ratio of the depolymerization upon the polymerization will be fixed. So this means that if the polymerization rate of the positive end is higher than the polymerization rate at the negative end, this means the depolymerization rate at the positive end will also be higher. So in other words, the G-actins will be added at the faster rate at the positive end, but they will be removed at the faster end at the faster rate at the positive end as well. So let me rephrase this: the rate of addition of G-actin as well as rate of removal of G-actin at the positive ends, both of them will be higher than the rate of the addition or rate of removal of G-actin at the negative ends. So this means even if K on of the positive end is higher than the K on of the negative end, this will be same in the case of the K off. Okay. And that is why the ratio K off upon K on will be constant. Okay, so this ratio is constant in both of the scenarios. So the thing I want to explain here was that at the positive ends and the negative ends, they have different requirements. So due to the different conformations, so due to the conformational differences, the addition, the concentration which is required, okay, for polymerization at positive end, that will be lower than the concentration that is required at the negative end for polymerization. So let me give you an analogy. Let's say that the positive end is a studious person with an high intellect, while the negative end is an average person with a mediocre IQ. So now the so if you want to explain a certain concept to both positive and negative end, to a studious person or to an average person, you will have to give different analogies to both of the person. The studious person will get the concept easily compared to the average person, because that person already has a high intellect. So this means that requirement of the critical concentration at positive end, it is less than the requirement of the critical concentration at the negative end. So the explanation that you have to do to illustrate certain idea or to convey certain idea, that will be less in the case of the studious person. That person will get it more easily compared to a mediocre person, and that is why this is same in the case of positive end and the negative end. The positive end has the critical concentration lower than the negative end, means even if, let's say that the value for the critical concentration at the positive end is, let's say, 0.6 micromolar, and at the negative end it is 1.2 micromolar. Okay, so having numerical values will help understanding this better. And the critical concentration of the G-actin in the cell is 1 micromolar, means the availability of G-actin is this in the cell, but the critical concentration needed by the negative end is higher than this critical concentration, right? This means that the G-actin will not be added on the negative end, but when we look at the positive end, it only requires 0.6 micromolar availability of the G-actin, and in the cell 1 micromolar of G-actin is available. 1 micromolar is more than 0.6 micromolar. So means we have sufficient amount of G-actin so that it will be added to the positive end. So we see that the addition of G-actin is seen at the positive end, but due to the higher critical concentration of the negative end, the G-actin won't be added at the negative end, and this is known as actin treadmilling. Okay, the value of critical concentration at negative end being more than the value of critical concentration of positive end, this phenomena is known as actin treadmilling. Okay, let's say if in the cell, so the critical concentration for at negative end was 1.2 micromolar, and at the positive end it was 0.6 micromolar. And let me give you three scenarios. Critical concentration of cell is 3 micromolar, critical concentration of cell is 1 micromolar, and critical concentration of cell is zero point 0.3 micromolar. Okay, so in the first scenario, when the G-actin concentration is 3 micromolar, the polymerization will be seen at both of the ends because the critical concentration, the requirements of the critical concentration is met at both of the ends. It is the amount of this G-actin which is available in the cell, it is sufficient for both the positive ends as well as the negative end. So in this case, in the case one, we see polymerization of G-actin to F-actin means the filamentous actin is being formed. Okay, we can also say that this is in the log phase. Okay, in the second case where the concentration is 1 micromolar, we have already discussed this case that at the negative end the concentration is higher than the concentration of the G-actin which is present at the cell. That is why at the negative end the depolymerization will be seen, and at the positive end the polymerization will be seen. So we can say that this is the steady state scenario. The second case is represented, it is representing steady state scene, means the number of some G-actins are being removed. We can't exactly say that it is representing steady state, but we can say that it is representing a stage where some G-actin molecules are being removed from the negative end while some G-actin molecules are being added to the positive end due to the differences between the critical concentration of both of these ends. In the third case, where the critical concentration of cell is much less than both of the critical concentration, the concentration at negative as well as the positive end, in this case, depolymerization of F-actin will be seen. Means disassembly of F-actin will be seen. So this is exact opposite of the first case. So depending on what concentration of G-actin is present in the cell, whether the G-actin will be added to positive end or negative end will be decided. Okay. So I hope the concept of actin treadmilling is clear to you all. Let's now discuss about the different factors that affect the formation, the process of formation of the actin filaments. So ionic strength has effect on the actin formation. The more is ionic strength, you will see that it affects the polymerization in a positive way. Means the higher is the ionic strength, higher will be the polymerization rates. Okay, so it is directly proportional to polymerization. Then there are there are certain factors, the certain proteins which we call as the accessory proteins like formine or ARP2 or 3, which are going to help which are going to either help or help in the polymerization or help in the depolymerization. I have a chart for different types of the proteins that are going to affect the polymerization of T or the depolymerization of F-actin. So we'll discuss this in a while. Then the third factor which affects the formation of F-actin, which we did not touch yet, is the presence of ATP. So even though this process is doesn't require ATP, but the as we discussed earlier in the start of the class, that G-actin has a pocket for ATP binding. So either ATP or ADP can be bound to G-actin. So there are two types of G-actin subunits. The subunit which is ATP bound, which is also known as T form, and the subunit which is bound to ADP, which is also known as D form. So either it can be associated to ATP or ADP. So association of G-actin with ATP increases the polymerization rates. Okay. So even though you do not necessarily need ATP hydrolysis, the energy which is derived from the ATP hydrolysis to start this process, but if ATP is present there, it is going to increase the polymerization rate. So these are the different factors that are going to affect the formation of the F-actin. So let's now discuss the different types of proteins that are going to help in the uh, polymerization or depolymerization. Okay. So the very first protein is known as formin. So formin, it attaches itself to the growing end or the positive ends. Let me zoom the picture a bit. So they associate themselves with the positive ends and they help in the polymerization. So let me write it here. So they help in the polymerization. Okay, so formin, this protein attaches to the G-actin at the or F-actin at the positive ends. So it actually attaches itself to the monomers as well, and so this is going to also help in the nucleation, and thus it is going to help in polymerization. Okay. Then there's a different protein known as ARP2 or 3 protein. It is also known as the branching protein. So this is going to call, this attaches itself to the negative end of the F-actin, and it helps the different F-actin filaments to attach themselves to each other. Thus, this is also known as the branching protein. So this forms the branches, the branch network of the actin filaments. Here you can see this green colored protein is attaching itself to the negative end, and that is how it is going to form a branch network of the F-actin. So this is also known as the branching protein. Then there are two proteins known as profilin and cofilin. They are also known as actin remodeling protein. So now, during the different cellular events, it doesn't necessarily mean that the G-actin polymerization or F-actin depolymerization is always going on. There are certain cellular events where you just need to tweak the actin filaments a little bit, and then you're ready to go. You're ready to do the cellular functions. So this is what we refer as the actin remodeling. Means the actin filaments, they are not depolymerized totally, but they are changed a little bit, and that is what we call as the actin remodeling. So in this process, two types of proteins help. Those are known as profilin and cofilin. So profilin, this protein, it binds to the monomers, the G-actin monomers, which are ATP bound. Okay, so always remember, profilin attaches itself to the G-actin, which is bound to the ATP. So ATP associated G-actins, they are recognized by profilin, and once profilin attaches to those G-actin monomers, it increases the polymerization rate. So you can also say that profilin is the protein which is going to increase the polymerization, or which is going to form in the which is going to help in the formation of the F-actin or the filament assembly. On the contrary, cofilin it attaches itself to the ATP bound G-actin. Okay, and when it attaches itself to the ATP bound G-actin, so now this ATP bound G-actin is not free, and it can't be assembled into F-actin, and this is how cofilin indirectly helps into the depolymerization event. So due to the attachment of cofilin to ATP, so cofilin is also known as ADF. Okay, ADF cofilin. So this ADF cofilin protein, it attaches to ADP G-actin, and it it accelerates disassembly. So due to that, so let me show you a schematic. So this is G-actin, So we already discussed that if G-actin, ATP associated G-actin monomers are there, they will increase the polymerization rates. Okay, so to this, so let's say that there is one other G-actin molecule. So this is G-actin. So this is ATP associated G-actin. So once ATP associated G-actin, to this ATP associated G-actin, the profilin binds. So this is profilin. So to this profilin binds and it helps in the polymerization. Okay. Upon the ATP hydrolysis, this F-actin now will look something like this. So we already know that the G-actin can be associated to both ATP as well as ADP. So let's say that ATP hydrolysis is only occurring at the this G-actin. Okay. So the rest of them are the ATP associated G-actins. So once ATP hydrolysis occurs, so now this ADP, this ADP associated G-actin will detach itself from the F-actin. Okay. So this will result into disassembly. So those are ATP associated G-actins. And now this ADP associated G-actin is free. So to this ADP associated G-actin, cofilin will go and bind. So this is cofilin. And due to the binding of this ADP associated G-actin, the depolymerization or disassembly of F-actin is accelerated. So now due to the binding of cofilin to this ADP actin, this will result into failure of this ADP actin. So now inorganic phosphate cannot be added to this ADP and this cannot be converted into the ATP actin, and due to this phenomena that the conversion being not the cell which is not being the cell is unable into the conversion of ADP actin to the ATP actin, so now the polymerization will not be seen. So this is how it is indirectly accelerating the disassembly. This is also known as sometimes it also directly fragments the F-actin, and that is why it is also known as chopping or savoring molecule. Okay, so cofilin results into depolymerization, while profilin results into the polymerization of the G-actin. Okay. Then there's other proteins such as tropomyosin. So tropomyosin binds to the other accessory proteins and it helps attachment of actin filaments to other filaments. Then there are certain proteins known as capping proteins which binds to either which capping proteins bind to positive end, but a different type of capping protein is known as tropomodulin. It binds to the negative end. So the binding of capping protein at the positive end, this prevents assembly or disassembly of the plus end. So it stabilizes the plus end. While the addition of the tropomodulin at the negative end also stabilizes the negative end. So remember those names, the capping proteins like CAPZ, they bind to the positive end while the tropomodulin binds to the negative end. Then there are proteins like gelsolin. They also known as savoring proteins. So they severs the filaments, means they chop the filament, and they usually bind to the first end after the positive end after chopping of the filaments. Okay, so there are here you can see alpha actinin as well as fibrin. So those proteins, they help in attachment of the actin filaments to the other actin filaments, while the filaminin protein, they also help in attachment of actin filaments to other filaments, but in a mesh like manner. Okay, so fibrin, alpha actinin and filaminin, they help in the attachment of actin filaments to the other actin filaments. But on the contrary, the a molecule like spectrin. It attaches the actin filaments to different surfaces like plasma membrane. Here you can see ARM. So here you can see the actin filaments. They are being attached to the plasma membrane in a star-shaped manner. So this is the work of the spectrin. So I covered all of the molecules. Yeah, I forgot to cover thymosin. So thymosin, this prevents assembly. So this, unlike profilin, which accelerates the polymerization of G-actin to F-actin, thymosin accelerates the depolymerization. So due to the binding of thymosin to the monomers, now those monomers are not free and they cannot go and form a F-actin. So those are the different types of accessory proteins that are going to result into the either increase in polymerization or decrease in polymerization. So with this, I conclude the lecture. Next lecture, we will learn about intermediate filaments and microtubules. Till then, see you. Bye bye.